We've already discussed the innate immune system. Now we would like to discuss the adaptive body defenses. A little wisdom from Al here. Education is not the learning of the facts, but the training of the mind to think. This is very true with the immune system. There are a lot of bits and pieces that you can learn as far as flashcards, but you really have to be able to put all these pieces together, just like with any other system. But the immune system, being systemic, running through your entire body, is a lot of layers to get through and to put together like puzzle pieces. So thank you, Al, and rock on. Adaptive defenses. We have already covered the innate defenses. We've talked about the first layer of defense, skin barriers, the skin mucous membrane. We've gone into the second layer of defense once we get inside that barrier, which are all nonspecific phagocyte action, those natural killer cells, the nonspecific inflammation, two antimicrobial protein sets, the interferon and the complement proteins, and that general nonspecific fever that runs through your body to help make the invaders uncomfortable and hopefully a little more uncomfortable than you. Today we are going to start on antigens. What are they? There's different types. Cells, an overview of what's going on in the adaptive defense system, which are B cells and T cells, and there are different types of B cells, T cells. And then we're going to spend the rest of the time talking about the B cells themselves, which are known as humoral immunity compared to T cells that are cellular immunity. So T cells go after cells that are infected internally. B cells work through the blood and the fluids, the humerus, with their antibodies. Adaptive defenses here. We are now into the specific, not the innate that's non-specific, but adaptive defenses are very specific. They are going to recognize and target very specific antigens. All right, they are looking for one little piece of one little bacteria or one little component of one little virus to be activated. So these guys are systemic. They're not restricted to the initial site. Okay, they can run all around the body. New to these as well, the adaptive defenses, is they have memory. Some of these B cells and T cells will live forever. There's a small fragment once you have an infection that will live on forever. The others will die, but they're there to recognize that attack again if it's the same virus, same bacteria, and the response will be stronger and it'll be faster, which is very cool. These cells are the whole reason that vaccines work. There are two separate overlapping arms I mentioned in the adaptive defenses. You have your humoral B cells, which are antibody mediated, and you also have cellular or cell mediated immunity. And these are the T cells that are going to attack our cells themselves. Okay, what's going on inside them compared to what's going outside of your cells. So in the interstitial fluid, blood, things of that nature. Our B cells. Humoral immunity, okay? These are the guys that produce antibodies, tons and tons and tons of antibodies. And the antibodies freely circulate their protein through all the body fluids. Okay, bind to target cells, whatever they are very specific for. That B cell will make antibodies to that specific component, a little piece of a bacteria, all right, and inactivate that cell or flag it to be eaten by macrophages or other munchy cells, neutrophils, dendritic cells, things of that nature. So it helps to inactivate, it helps to mark them for destruction. Remember, it even helps to mark for destruction via complement proteins. Remember, they are activated by antibody and help to poke holes in that bacteria, that MAC attack complex. So again, humoral immunity has extracellular targets outside of your cells. The T cells, the cellular immunity branch of the adaptive system, is our lovely little lymphocytes that attack against target cells. So say a cancerous cell or one of your self cells that's been infected with virus. These guys kill directly, all right? They will touch onto that cell with their T cell receptors and cause that cell to die. They can also act indirectly. They can release chemicals near the cell, okay, that are going to enhance inflammatory response, bring in other components of the immune system. help to activate other lymphocytes, really get those macrophages raging. So they work twofold. They can tell cells to die, and they can also up the ante on the immune response and draw on other components as well of the immune system. So again, cellular immunity is going to have cellular targets, cancerous cells, virally infected cells, anybody that's not looking right. So here is the outline for the adaptive defenses. We're going to talk about antigens. What the heck are they? There are complete antigens. There are incomplete antigens that need a little help to cause an immune response. We have many, many antigenic determinants on antigens, and I'll explain what that means. We also have self-antigens, okay, known as MHC proteins. And I'll let you know what those are. I've already touched on them a little bit when I said natural killer cells recognize proteins on our tissues, and if they aren't there, they'll kill them. It's when these guys, these MHC proteins, get downregulated and are not on the surface that the natural killer cells attack. Then we will go into the cells and overview. We're going to go a little more in depth now on those lymphocytes. We've always talked about T cells and B cells. Well, there are different types of T cells that have different jobs, and B cells can change as well. And we're going to talk a little bit more about those phagocytic cells as antigen-presenting cells. Remember, I said they can go back to the lymph node where the T cells and B cells hang out and present little pieces of antigen. That's how they act as an antigen-presenting cell. So this is another job that they have. And then the third topic will be humoral immune response, which is all about our B cells. We know how they're born, we know where they're born, but let's talk about how they get activated, how they differentiate into those antibody-producing plasma cells or into memory B cells, okay, giving us that immunological memory. We'll talk about different types of humoral immunity. There's active and passive. Okay, active is when you're going to be making them antibodies yourself. Passive is when you might be getting them from someone else, say from mom to baby. And then we'll talk about the different types of antibodies. There are several different types. These are just 3 different shapes that you have. And for this monomer here, there are actually 3 different types there. So there are a total of 5 different types of antibody, each having their own job and located in different areas of the body as well. So let's get cracking on topic 1. What the heck are antigens and the different types? Antigens. You'll sometimes see it shortened as AG for antigen. Antigen is actually a put together of antibody generating. So anytime you see antigen, this is something that will generate antibodies against it. There's all different types of antigen that can create antibodies. So antigens are substances. They can mobilize adaptive defenses and provoke an immune response. So you can better believe that antigens are made out of proteins, protein carb complexes that we don't make ourselves in our genetic makeup, something we're going to recognize as non-self. Targets of all adaptive immune responses are going to be antigens. Most of them are large, complex molecules, again, not normally found in your body. Okay, they are non-self. What happens if we recognize self and create an immune response? Antigens that are our own. Yep, you're absolutely correct. Autoimmunity. We'll talk about a few of those when we go over some disorders. All right, I mentioned there's different types of antigen. One type is a complete antigen, so all by itself, it can cause an immune response. Complete antigens have a function property known as immunogenicity, and that just means that they have the ability to generate an immune response all by themselves. Okay, ability to stimulate proliferation of the specific lymphocyte that recognizes that little piece, that antigen. They also have a property known as reactivity, and this is the ability to react with activated lymphocytes or antibodies that were released by B cells. Right? So they can connect all by themselves. They don't need any assistance. Things like this would be foreign proteins, some polysaccharides, some carbs on the cell surface of a bacteria that don't look like anything we make, certain fats, some lipid structures, nucleic acids that don't belong in sequence in our DNA, pollen, causing an allergic reaction. It's an antigen. Bacteria, definitely. Fungus, these cute little fungi causing ringworm here. It's actually not a worm, it's fungus. And yes, even viral particles are recognized as complete antigens. They will activate T cells, B cells, things of that nature. So complete antigens can do it all by themselves, kick off our immune system. Haptens are known as incomplete antigens. Okay, so haptens themselves, there's these cute little things here. These are considered haptens. They're small molecules. They cannot cause an immune response by B and T cells by themselves. Okay, it's not going to happen. Small peptides, little pieces of nucleotides, no. But they can become immunogenic, causing an immune response if they attach to your own body proteins. And then that combination is large enough to potentially be recognized by T cells and B cells if that's the specific sequence they're looking for and they were trained for, and cause an immune system response. Now, this is bad, right? Because those little haptens attach to your own protein. So now you're attacking yourself. Prime example, poison ivy. All right, the oil from that plant attaches to self proteins and elicits an immune response. If you have allergies to animal dander, pet dander, it will bind. It is a hapten. Detergents causing that itchiness on your skin, it's actually binding to the skin and then causing an immune response to self, and some cosmetics as well. So when these haptens get together with a carrier molecule, a protein of ours, they then become, wonder powers unite, a recognizable antigen. And this response is known, there's several different types, as a hypersensitivity, okay, an allergic reaction. And we're going to cover this in a little more detail in the next PowerPoint. So I mentioned antigens. Now let's talk about antigenic determinants. Only certain parts of antigenic determinants of an entire antigen are immunogenetic, meaning not the entire, if this is a bacteria, the entire bacteria is not recognized as an antigen, right? It's got a whole bunch of surface markers on it, just like our cells have hundreds of different markers on the surface. And the T cells and B cells are looking for very specific little components. So on the surface of this bacteria, there'll be different glycoproteins or different proteins or different receptors. And antibodies and also the T cell receptors are looking for these little bits and pieces on the surface. These are all antigenic determinants, different shapes, different sizes that'll activate different T cells and B cells. So against bacteria, we can have many different lymphocytes activated and creating antibody and attacking these bacteria because they have multiple activation points on their surface. Those are known as antigenic determinants. If you think of an example, transfusion of the wrong blood type, you're not attacking the whole blood cell. It's actually recognizing just the A marker on the surface, that protein, or the B, or both if you're type O blood. Those are antigenic determinants. So most naturally occurring antigens, again, just like I showed you, have multiple different antigenic determinants on the surface. This antigen showing here has a little triangle, half circle, little tab A, slot B. These are the antigenic determinants. So like I said, it can mobilize several different types of populations, form multiple kinds of antibodies against them, and your macrophages, your antigen-presenting eating cells and DCs, all recognize the end component of these antibodies to go after them. Large or very chemically simple molecules like plastics, like metal, have little or no ability to generate an immune response. So think about somebody who has screws or plates to hold their bones and joints together. There's no immune response to those because it's just similar metal, very smooth. It doesn't have all these different proteins and different glycoproteins on the surface like bacteria do or virus. You can use plastic in your body as well. When you're doing those valves that I showed you for the heart, those plastics, it's not going to create an immune response. So that's a nice component to use instead of a true pig valve, which would have all these antigenic determinants. Self-antigens. I mentioned this as well, right? We have self-antigens like the MHC proteins that I mentioned earlier, okay, like the A and B proteins on blood. So these are protein molecules on the surface of cells that are not antigenic to self, but antigenic to others. So these are things that I have that maybe won't work with you, and it's going to make a big difference in transfusions, a big difference in tissue grafts, transplanting kidneys, other organs, things of that nature. So I think I've already given you an excellent example that you're going to come up with right now. That's right. Your ABs, your RH blood antigens can cause an issue. And what that is, another example for that is the MHC glycoproteins that I mentioned earlier. MHCs are major histocompatibility complex, MHC, coded by genes. So these are proteins, and they are unique to every individual. All right, so my epithelial cells have certain MHCs on the surface, my muscle cells, my nerve cells, my leukocytes, a little different than what you have. Right? These are the things that when they're downregulated, that's what those natural killer cells can go after if you're not expressing these on the surface. And what they do is they're cute. They have a little groove in them, so it's kind of like a hot dog bun. And in the groove, we lay the hot dog or a self or foreign antigen that might actually be inside. So if you're not infected, you're just putting out little self hot dogs in that hot dog bun, that groove, and saying, hey, I'm kosher to the T lymphocytes, no problem. Hot dog kosher. I didn't mean to do that, sorry. Ha! But if you're infected with virus, you take some of those components and you present them out on the surface, and that's how you let those T cells know I'm infected with something that shouldn't be here. And the T cell will tell you to undergo programmed cell death. All right, so these MHC proteins are very important in creating an immune response. Question time. Which of the following materials or compounds would be the most highly immunogenic, so really generate an immune response? Non-self antibody, plastics, a hapten all by itself, or an antibody that you made? What do you think? Take your pick. Here we go. A non-self antibody, right? If it's self antibody, you're going to recognize it as self. If it's a hapten all by itself, it cannot create an immune response unless it binds to another protein of yours to create that complex with the hapten. Remember, plastics we don't create an immune response to. Non-self antibody. So this is an antibody from somebody else, not part of your genetic makeup to make this antibody. You're going to attack it. And yes, antibodies can attack antibodies. Pretty cool. The presence of blank proteins makes it possible for our immune system to differentiate between our cells and those that are foreign. Okay, so is it antigenic determinants, MHCs, those haptens again, or perhaps antibody? All right, take your pick, and here we go. MHC. It's the presence of MHC proteins on the surface that make it possible for our immune system to differentiate between our cells and those that are foreign. They'll recognize other components, but if they've got our MHC on the surface, that's the secret handshake. All right, and they'll be located on different cells. And I'm going to blow your mind later when I tell you there's different types of MHCs. Blank become antigenic if they attach themselves to larger proteins. I know y'all got this. It's not the pyrogens that cause that fever. All right, you know this. It is those haptens, those cute little tiny molecules that when they attach to a self-protein, like the oil of poison ivy to our proteins, our epithelial cells, on the skin, we are going to have a nasty reaction. All right, that is the end of this lecture component. The next video will be covering the cells, doing an overview of lymphocytes and those antigen-presenting cells, which we've already named, like dendritic cells and macrophages. But we're going to give them another job where they actually present, and we'll talk about that in just a little bit.